Hello guys, welcome to my channel Plan Science for You. In this video, we are going to talk about biometric techniques in plant breeding. We are studying plant breeding using field experiments. Okay, you have to know about different techniques used in plant breeding and in agronomy also. So we'll talk. It will be a video series where will be talking about different experimental designs, different introductory statistics methods, different aspects or different methods to analyze statistical data, etc. So subscribe my channel, and if you like the video, share with your friends, and also join our Telegram channel, Plan Science for You. In this video, we'll talk about introductory statistics. That means different types: the first order statistics, second order statistics, third order statistics, etc. In our next video lecture series, we'll see different types of experimental designs like a randomized block design, completely randomized design, lattice design, augmented design, etc. After that, we will see different applications, or we can say the components of biometrical techniques which we will further use. After that, we will see polygenic variations, how polygenic variation is measured. For that, what are the methods are available, like Metro-Gilp analysis, discourse statistics, and all that. In our fifth lecture, we will saw the determination of the yield components, how we can determine the yield components using correlation analysis, path coefficient analysis, and also the discriminant function analysis. In the sixth lecture, we will assess the gene action, what are the gene action is carrying in the field between two varieties, what is the gene action is going present, what are the breeding methods we can use using the analyzing the gene action using diallele cross approach, triallele, partial diallele, tetraallele, etc. In the last video lecture, we will saw about assessment of varietal adaptation. What are the different models which are available to assess the varietal adaptation, like Everhart-Russell model, which is very important, Finlay-Wilkinson model, Parkins-Jeans model, and Freeman model. Okay, so let's start today's topic, which is about introductory statistics. If we see the definition, what does statistics mean? It's actually a branch of applied mathematics where we apply the mathematics to deal with different numerical data which we got from the field, or collection of data, presentation of those data, and analysis and interpretation of that data to get a conclusion. Okay, so what does the statistics means is actually the branch of applied mathematics which deals with different aspects of analyzing a data and get to a conclusion. Okay, what we can do, we can use those statistical methods to analyze genetics analyze our field data in genetics and plant breeding, and in agronomy also. We can divide those methods actually into two different parts. Firstly, different statistical methods which we will going to be used in the field for analyzing our data collection, presentation, and interpretation of the data. And second one is the experimental designs. What exactly we will see in case of in the field. There is a different variability also present in the field. So how can we allocate our checks? How can we allocate our varieties to get a better representation of the whole field, or the better capacity development of the actual variety? Then if we talk about degrees of statistics, we can divide statistics into 3 degrees depending on which method or which parameter is used to analyze the statistical data. We can divide it into first degree of statistics, second degree of statistics, and also third degree of statistics. In case of the first degree of statistics, it is actually based on the mean value. Mean means average value. Okay, it is very robust and also reliable, and it provides a high degree of precision and it can be used in case of generals and mean analysis, heterosis and inbreeding depression studies, combining ability effect studies, meteorological analysis, stability analysis. Don't worry about these names. We will talk everything in our foregoing videos, so keep on watching. Don't worry. In the second degree of statistics, second order of degree of statistics, which is based on variance and covariance estimate value, which is actually not too much robust or reliable as we can see in the first order statistics, because it is based on variance and covariance data. Its precision we can increase by simply increasing the sample size. The more number of sample we have in our hand, we can have more data. We can have a more uh, reliable and robust interpretation. Or we can use in this modern era, we can use of computer, we can analyze data more better. It can be used second order statistics we can use in case of correlation coefficient, path coefficient, discriminant function analysis, discourse statistics, heritability of the analysis, heritability of the trait analysis, estimation of genetic component for diallele, partial diallele, where we use variance and covariance data to estimate its result. Statistical data is mainly used for, we all know that this is a polygenic analysis. If we see in case of the oligogenic traits, like which are the qualitative traits, okay, we can distinguish them into discontinuous variation in is seen, and we can distinguish them into different classes like a red flower and a white flower. Nothing is in between. But in case of a polygenic trait, we can see the weight of a fruit. It varies from 60 to 70 gram, and there is 60, 61, 62, 63. There's a continuous variation is there. But in case of an oligogenic trait which is controlled by more than one or more than two number of genes, which is actually a qualitative trait, and it will exhibit a discrete variation, or we can say is a discontinuous variation. It is easy to classify, and we can interpret those data using frequencies and ratios easily. Here we use a very important terminology, that is the degrees of freedom. What does it actually means? It means about number of independent comparisons, how much of freedom we have in terms of independency. We can say it as a degrees of freedom. We can understand and this about this formula, like degrees of freedom, we can say it as n minus 1, where n is the number of classes. If say there is a four place and there is a four people, 1 2 3 4. When the number one pupil goes, you see there is a fourth chair is empty. Okay, but he choose number one slot. When the number two pupil goes, he sees there is only three seat is empty. He sits on here. Suppose here. When the number three pupil goes, he has only the two seats empty. He can choose any one of them. He sits like here. For the fourth person, there is no choice. There is no degrees of freedom. There is no degrees of freedom. He have to sit here. There is no any empty seat there and there, only this chair. So what we can see here, that there is a degrees of freedom up to n minus of 1 classes. This is defined as degrees of freedom. Suppose we have we see that one example which we use in case of monohybrid cross. What we'll see, that F1 ratio is of red and white flower is 3:1. That means there is a 3:1, two classes, means there is a degrees of freedom will be 2 minus 1, that is 1. In case of dihybrid cross, it will be F2. In F2 we saw the ratio is about 9:3:1, that is 4 classes. Then the degrees of freedom will be there 4 minus 1, that is 3. Then we see, if we see about the chi-square test, which is also very important, and it is was developed by one important scientist, that is the Carl Pearson. Okay, it is to test the significance of difference between the observed data and the expected frequencies or ratios. Suppose there is a 3:1, we can expect these ratios for an F2 in monohybrid cross, but if we get the result is 2.18 and is to 1.05, 1.05 or something like that. If we observe data is this and our expected data is this difference, there is a difference, is this difference is significant or not, or we can say it is equals to 3:1. We can know using this chi-square test. The formula you should remember, that is the formula is the observed frequency, observed value. O is the summation of observed value minus E is whole square by expected value. That means chi-square equals to summation of O minus E the whole square by expected value. Okay, the calculated x-square chi-square value is compared with the table value. There is a table value we will see of the chi-square at the 5% level of significance at n number one number of degrees of freedom. N is the number of classes. Like in case of there is a two class 3:1 in monohybrid class, there is a two class. Then degrees of freedom will be 1, and the we will check the degree level of significance at 0.05%, 0.05, that is 5% level of significance. Like here, this is the significance level, that is 0.05. We will see in case of monohybrid causes the degree of freedom is 1, and we will see here there is a value is 3.841. Okay, this is the table value. We will see this about the table value. Then after utilizing the formula, that is the chi-square equals to summation of O minus A whole square by E, we can calculate the chi-square value, and it is the chi-square calculated value, and we can also find the tabular value from the table calculation. And if we see the chi-square tabular value, calculated value is greater than chi-square tabular value, we can say that there is no significant difference is there. That means we can say our expected value, our observable value, that is 2:18 is to 1:05, it is equals to 3:1, which is our expected ratios. Okay, that means there is no significant difference is there, they are equal. If the calculated value is less than the table value, then we can say there is a significant difference is there, and we can say that 2.8818 and 1:0.5 is not equals to 3:1. Our expected our observable frequency is not equals to our expected frequency. Okay, then in which case we can apply the chi-square test for the estimation or testing of the segregation ratio, which we have seen here. For detection of linkage, if there is a presence of linkage or not. If there is a presence of linkage, there is a bit diffraction in the segregation ratios. We will saw the ratio from the given data. We will analyze them. If the difference is significant, then if there is a significant difference is there, then we can say there is a presence of linkage. We can use this method for analysis of gene expression in case of population genetics, gene frequencies in case of population genetics. What are the limitations? Firstly, it is all applied only to the numerical frequencies. It cannot be applied in case of ratios or percentage data, and it also cannot be applied to distribution in which the frequency of any class is lesser than 5. Okay. As we have discussed earlier, the polygenic traits. What are the polygenic traits? There is a continuous variation is there. There is no distinct classes, and it will be controlled by several genes. And we can say we can use the metric measurement for our analysis of polygenic traits. What are the simple measurements we can see here? There is a simple measurements of variability, test of significance, analysis of variance, covariance, regression analysis, and correlation analysis. We will see one by one. So in case of quantitative character, we simply use some simple measurements for measuring the variability. You can see in the central tendency measurement, we can use the mean value, median, average, and mode. Where suppose in a class in mathematics, some students got 25 to 89 numbers. Okay, there is a range of numbers. Okay, so we can establish them in a average value. We can establish them in the average value to assess the performance of the students. Then we can use a central tendency. We can make their averages. Okay, we simply make, there is a suppose a 45 student, so simply we can make their average, and we got to go. After that, we can calculate the measurements of dispersion. How dispersed is the number? How dispersed are the values? How uh, how the degree to which the numerical data tend to spread about the mean value. Then we can see to analyze it using the range. Suppose that in a class, it is the difference between the lowest and the highest value. Okay. Suppose there is a highest number is 89, where the lowest number is 10. Then the range will be highest number minus lowest number. 89 minus 10, that is 79. Okay. Then if we come to standard deviation, is the actually the variation, root over of variation, and it actually tells the uh, square root of variation. That means square root of arithmetic mean of all squares and of all deviation measures from the mean. That means you can simply just say that standard deviation is equals to root over of variance. Okay. From here we can also say that square root of variance equals to square root of SD. If we see in case of the standard error, which is we can say this is the measures of uncontrolled variation present in the sample. In a sample, there may be some uncontrollable variation is present, like in the field condition. Some part there is more fertile, some part there is less fertile. Okay, so some uncontrollable error may occur, that we can discuss about the standard error, and we can calculate the standard error using the formula standard deviation divided by root over of n, where n is the number of observation in a sample. Okay. Then we can see the coefficient of variation, that is the CV. We can calculate using the standard deviation by uh, x. That is the mean of a sample. X is the mean. Okay. One simple conclusion we can say, that is the coefficient of variation. If it is the more, then there is the sample is more variable. Okay. We can also use the test of significance. Okay. Where we can use the null hypothesis, which was given by scientist Fisher. Okay. What it states that that sample result which we will get calculation will be equals to population parameter. Okay, if there is a difference, then it will be, if it deviate, then it is due to chance only. Chance, that is environmental error is there. Okay, so there will be sample result equals to the population parameter. Then we can compare between two means. Okay, for analyzing the test of significance, we can compare between two means. If the number of sample is large, that is about up to a large number of sample is there, there is more than 30 number of sample is there, then we can use the Z test. If the sample number is less than 30, then we'll use T test. For comparison of several means, there is a several means. Then we can use the F test, which is we can use like F test is used when there is several mean is there, and we use it in case of ANOVA, that is analysis of variance, where we divide total phenotypic variance into genotypic variance and environmental variance. We divide the data and we go for analysis of variance. So in summary, we can tell that T test is used for less than 30 sample, Z test is used for greater than 30 sample. These two are used in case of, or there is a two mean, but in case of several mean we use F test. And this analysis F test is F value analysis of F value, which is based on ANOVA. Okay. As we have discussed, that analysis of variance, that is the ANOVA, it splits total variance into different components. Okay. It use replicated data, like it divides the total variance in two major parts. Like in case of VP, there is a total phenotype variance, which will be equals to VG. Okay, genotypic variance and environmental variance. We use replicated data to analyze the variance. Okay, in case of ANOVA, in case of analysis of covariance, ANOVA, we can say is ANOVA. In case of in this case, it splits the covariance. Okay, CV. It splits CV into two variables. CV of two variables into different components. If there is a two variable, that is x and y, x CV and y CV, we divide them into different components. Okay, we will see later on. Here another important terminology, that is the regression analysis. We can define as a B. It represents the average functional relationship between two or more variables. That means if we see there is a two variable is present, like one is a nutrient, which we can say is x1, and another one is the irrigation, which is x2. If we increase the nutrient, there will be increase in yield. If there is an increase in irrigation, there also will increase in yield. Okay, so you can say that yield component is dependent on x1, x2, like different factors. This functional relationship we can define as a regression analysis. Okay, like that way, if we can say that nutrient and irrigation, if we increase them, there will be an increase in yield. So we can say they are positively correlated. That means there is a mutual relationship between two variables. It may be positive or it may be negative. Okay. In case of if we increase, there is a increase is there in the another dependent variable, then we call as a positive correlation. We can express in case of, in that we can, like in a graph, we can express that if there is a one variable is there, another is a y. If we increase x, there will be an increase in y2. That means there is a positive correlation. But if we see there is a negative correlation, okay, then if we increase x, there will be a decrease in y. That means it is a negative correlation. But if there is a no effect of one variable to another, there will be an equal straight line, or you can say it is an unrelated correlation will be there. X, it is a y. Okay, so uh, regression analysis tells us about the functional relationship between two or more variables. That means they are dependent or not. If dependent, if there is a relation, there must be some positive relation, there must be some or negative relation. How, which type of relation is there, correlation is there, we can express this in terms of correlation analysis. Here we can see a comparison between correlation and regression analysis. Uh, in case of correlation analysis, we define it as a R, and in regression, we define it as a B. Okay, small b. In case of correlation analysis, it is about mutual relationship, and in regression analysis, it is about functional relationships. In case of correlation analysis, there is no involvement of cause and effect relationship. And in case of regression effect analysis, there is involvement of the cause and effect relationship. Okay. In case of correlation, then it tells about how much it is correlated. But in case of regression, there is a relation between one and one value. Okay. Uh, in the fourth point, what we see, there is a express measurement. In case of correlation, it is independent, but in case of regression, in the unit of measurement of variable की जो यूनिट है उससे मेजर करा जाता है. But in case of regression, the relationship is studied is a linear fashion. But in case of in correlation, it is linear fashion. In regression, it is linear and collinear both. In case of correlation, it measures actually degree and direction of the relationship, whereas in regression, it analyzes the degree of dependence of one variable to the another. Thank you. We will talk in the next video, where we will discuss about different experimental designs which we will use in case of our field experiments. So if you like the video, subscribe my channel, share with your friends. Thank you.